Hello students, this is biology and I am Saurabh Mukherjee. Today I am talking about the two structural configuration of a protein, that is domain and motif. But before the discussion my topic, so if you are new in my channel, then subscribe my channel and press the bell icon. At first, I am talking about the domain of a protein. So basically, there are various types of domain present in the protein, like DNA binding domain, catalytic domain, linker domain, and many other domain like transmembrane domain, activator domain, repressor domain, so many others. So basically, domains are as short as 40 to 700, 40 to 700 amino acid long. Various transcription factor contain many DNA binding domain. Basically, the domains are basically DNA binding domain contain a variety of structural motifs that binds to the major groove of a DNA. Only, that is TBP protein, that is Tata binding protein, only bind to the minor groove of the DNA, but otherwise all domains or the domains of the protein is mainly bind in the major group of DNA via non-covalent interaction. Here I will showing about various domains of a protein. So this is a helix, this is a repressor protein showing two DNA binding domain, one is here and another is here. So this is the DNA binding domain. This DNA binding domain found mainly in the protein and bind to the major group of the DNA. This is a helix turn helix motif. So various DNA binding domain contain motif like helix turn helix motif or zinc finger motif. So or zinc finger or motif, which I discuss in next slide. So there are various types of domain. One is the catalytic domain of a protein. This domain mainly found in the enzyme, and another is the transmembrane domain of the protein. This domain is found in protein that span the cell membrane, and it is typically consists of a sequence of hydrophobic amino acids. This slide, basically I am talking about the motif of a protein. So what are motif? Motif are typically short amino acid sequence, that is 10 to 20 amino acid sequence, which are conserved sequence found anywhere in the protein and associated in the specific function. So motif are the short conserved sequence found in the anywhere of the protein. So there are various types of motif present in the protein. One is the helix turn helix motif. This is a lac repressor protein containing helix turn helix motif. So this is a helix turn helix motif. Next is, this is a protein containing helix loop helix protein. Next is leucine zipper motif. So this is in this motif every seventh position of the protein contain leucine. So this is leucine. Every seventh position of the protein contain leucine. And the next and last is the zinc binding or zinc finger motif. So there are two types of zinc finger motif. One is so this is one type is C2H2, where Zn bind to the 2-cis residue, that is cysteine, and in here is histidine residue. So this is called the C2H2 type zinc finger motif, and another type is C4 zinc finger motif, that is Zn ion bind to the 4-cis residue, 4-cis cysteine residue. So this is all about the types of motif which I discuss here. So thank you students, all the best.